first of all, let me thank you for the opportunity to present our recent work on using some sort of rule or some sort of conservation law for deriving the entropy production in non-equilibrium systems. So that's the outline of my talk. I will present a brief motivation for the new approach. And then I will explain what is the variance rule. And I will show you applications to an application to derive the heat produced by a red blood cell. So regarding non-equilibrium steady states in general, there are two ways to produce non-equilibrium steady states, either because you exert a time-dependent force by, for example, changing in time stochastically a control parameter that here I show as lambda of t, and then this force derives from a potential. So the force would be conservative if the control parameter is time independent. However, because the time control parameter changes with time in a random way, let's say, you can reach a steady state. Other types of systems are those in which you can impose a net current of any conserved quantity by introducing a gradient of an appropriate potential across boundaries. So I will focus in this talk on this first type of non-equilibrium steady systems as the so-called force, or if you want, FNES, non-equilibrium steady state systems as simple models on which we can apply, or simple theories in which we can apply them to experimental data. So let me start just by putting in perspective what I'm going to show is, let me start with a very simple setting that was shown actually in the previous talk. You have a particle that is moving in this time-dependent potential, stochastically driven, and then the system is subjected to thermal noise. Now the variable describing the position of the system is x, and here you can see in the slide the mobility mu and the diffusion constant d, which is related to mu by the fluctuation dissipation relation. The noise is white noise, and in this case, if you know the force as how it changes in time, then you can measure the entropy production rate, or the entropy production, let's say, if you just integrate the Stratonovich force times velocity. And actually, this method of this way of measuring the heat or the entropy production rate, if you want, is the one that has been implemented usually in numerical simulations, but also experimentally there are ways to extract the heat produced in a non-equilibrium steady state by just directly measuring the relevant variables. So here I show you in the bottom of this slide an example of an experiment that I like a lot. Was done almost 20 years ago by Sergio Ciliberto and Garnier in Lyon, and what they did was to measure the heat fluctuations in a resistance, across a resistance, in an electrical circuit when you drive it with a current. And what you see is that not only you can measure the entropy production rate, but you can also measure its fluctuations. And its fluctuations satisfy a fluctuation theorem, which is what you can see in the center of the slide in this sort of box. So I don't know, for some reason doesn't work my cursor, but I don't know if it doesn't work my cursor, but I hope you see clearly in the center of the screen, that's a relation. But here what I'm talking is not about the fluctuation theorem, which is a sort of symmetry satisfied by the heat produced by the system, but it's a much more, or apparently simple question is if we can measure the average entropy production in a non-equilibrium steady state. And here in this integral in the center of the formula, what you see is an average over bar, which means this sort of time average, in such a way that the entropy production rate is time independent, is constant. So the question is, how can I measure this entropy production rate sigma if I sometimes cannot access neither the forces or the velocities? Because it's clear that if from a system you know all the velocities and force acting on all your particles, then clearly you can measure the entropy production rate. And this is what I'm aiming to answer, and to show you that in the case, for example, of a red blood cell, where we even we cannot measure the net force acting on the membrane of the red blood cell. We cannot measure the speed of the membrane of the red blood cell. Still, we can get an estimation of the entropy production rate. So there has been a lot of activity on this important problem, and actually most of the work has been developed by Udo Seifert, Andre Barato and co-workers during the years, and this has spurred a lot of activity. And basically what they say is that if you can measure a current and you can measure its average value and also its variance, then you have an inequality at finite times that, and also asymptotically for very long times, that if you measure the average square of the current divided by the variance, then you have this lower bound for the entropy production. Now, the problem with this inequality is that if you apply it experimentally, and we did this with Udo and Patrick Piazzonk a few years ago, when Patrick visited me in Barcelona, we did some experiments on a stochastic switching trap, and we observed that if you measure these, if you try several currents, non-equilibrium currents, for the system, which depend on what you define by force and displacement, you can construct two different types of current. Then what you can see, and here is shown in the plot in the right bottom part, that the ratio between the variance and the average work, which in this case should be bigger than 2, it is actually bigger than 2, but it is bigger than 2 by far, because if you look at the scale in y-axis, it's logarithmically. And, you know, if instead of 2, you get something like 4, 5, 6, 10, or even 100, 10 to the power 2, as you see in the plot, then you are off by more than 100%. So the question is whether there are methods, alternative methods, to find the entropy production rate without relying on a lower bound. And this is what I tried to do by introducing the variance sum rule. So let me motivate you why there is a need for a different approach rather than a lower bound. If you remember from second course thermodynamics, for example, if you take a thermodynamic system and you ask, okay, I start from an initial equilibrium state, and my system, for example, in this slide here is an hydrostatic system defined by pressure, volume, and temperature. Suppose that you do a transformation. Let it be an adiabatic transformation, let it be an isothermal transformation, but this transformation is irreversible. So it goes through non-equilibrium states. It doesn't go reversibly or quasi-statically. And at the end of your transformation, thermodynamic process, you get final state variables, pressure final, volume final, and temperature final that you see on this big circle on the right. And you ask, okay, given if I tell you the initial state, and now I tell you how is the protocol, so I tell you how I do this irreversible process from the initial to the final state, and I ask you what is the final state, then the answer to this question, what is the final state? Of course, you could say, okay, we have Clausius inequality, it's an irreversible process, and therefore the change in entropy you see on the right top of my slide, the change in entropy is bigger than delta Q divided by T. So if I measure delta Q divided by T, in principle, I should know delta S. But this is not true because there is an inequality. And actually, the way to know what is the final state of your system is that you need, besides the protocol, the precisely reversible process you are applying to your system, you need to have an equation of the state. If you have an equation of the state for your system, then immediately, if you know that the energy, for example, if it is adiabatic, the energy is if it is isothermal irreversible process, you know that internal energy is conserved. Always, if it is adiabatic, also the internal energy is conserved. But if you have the equation of a state, immediately, besides knowing the precise protocol you're applying, if you know the equation of a state, you know the final state of the system. The only thing you have to know is to apply the conservation of energy. So this is what we try to do in the variance sum rule. We try to go some sort of constraint reminiscent of the conservation, energy conservation, that allows us to know in the process during the stationary non-equilibrium steady state how much heat is produced and released to the environment. And here I show you in in a very summarized, synthesized way, what is the variance sum rule? Although you can find the derivations in the appropriate reference, you take the Langevin equation with a time-dependent force, you squared the equation, take averages, and this is very simple. What you get is an equality that tells you that the variance of the displacement times the variance of what they call the impulse, or what we call the impulse or cumulative force, which is the integral of the force, is equal to 2 dt plus a term s of t, which is called excess variance. Now, if you're in equilibrium, I anticipate you that in equilibrium, what we have is that s is equal to 0. And then immediately you have a conservation, like saying that the sum of the two variances, one of the displacement, the other from the impulse, is equal to dt. Now, if you are out of equilibrium, there is an excess term, s of t, that gives you the excess variance that is related to the entropy production. So why the excess variance is related to the entropy production? So here I show you an illustration of this total variance. The total variance is shown in this figure here. So the total variance is shown as the sum of the two variances, v delta x, v of the impulse. So the variance of the displacement in the illustration on the right is in color blue, or in blue, sky blue, let's say, cyan color. The variance of the impulse is in red. If you sum the two things, you get the red, which is the total variance. And this is in log-log scale. So I'm adding two things in log-log scale. So the shift in log-log scale is not twice, it's just a little bit, but that to illustrate the variance sum rule, and the dashed line is 2 dt. So the arrow that you see in that plot is the difference between the green and the dashed line. This is excess variance. And this excess variance that you see in the formula is just expression how much the time reversibility breaks down in the non-equilibrium steady state. Now, it turns out that if you know this excess variance as a function of time, this s of t, you can take the second derivative respect to the time, which is the formula you are seeing, and you get the entropy production rate. Now, it turns out that this entropy production rate for a particle that has an average speed v is equal to the first term, v squared divided by the mobility. This is Stokes friction term, plus one term, which is a second derivative of the mean square displacement with respect to time, evaluated at 0 time, plus a positive term, which is the variance of the force. So the last term is not anymore the variance of an impulse, which is an integral of the force, it's the variance of the force itself, which is a time-independent quantity. Now, you can see that in equilibrium, if you have an average, if you are in equilibrium, the speed of your particle is 0, then the sigma is 0, the speed of your particle v is 0. So actually what happens is that these two terms, the second derivative and the variance of the force, must compensate. But the variance of the force is positive, so the second term of the derivative is negative, okay? So this is a sort of compensation between the two terms, one positive and one negative, and these must have different sign. Of course, if you are in an equilibrium steady state, then the second derivative can be positive or can be negative. We don't know, okay? But this gives you actually the exact expression out of equilibrium. So we apply this to exactly solvables, well, a system that can be not only exactly solved, but experimentally realized. And that's a system that we studied a few years ago, also in collaboration with a student of Udo Seifer, Ed Dietrich, who came to Barcelona, and we implemented the stochastic switching trap, which is a trap that moves stochastically its position between two, inside water between two positions, and then introduces a steady state. This position of the trap that switches between two positions is experimental realization of that control parameter that I told you in the beginning, that it can be changing randomly as a function of time. Now, if you implement this stochastic switching trap, I won't give you the details. You can evaluate exactly the variance sum rule, all the terms entering in the variance sum rule. In particular, you can get the excess variance as that you are seeing in this expression squared, and from that you can get an expression for sigma. Now, you can test experimentally, and I show you here two realizations of the experiment with two amplitudes of the switching trap, and you see that the experimental points for different delta lambdas, amplitudes of the trap, fall outside the dashed line, which is 2 dt, and the larger the amplitude of the switch, then the stronger the non-equilibrium. Actually, the larger the excess variance and the deviation of the experimental data relative to the straight line. And here I show you what we measure for the entropy production rate. In experiments shown as red dots compared to the theoretical prediction, which is a function very simple. Well, it's not a complicated expression. It's shown in the box, and it grows proportional to delta lambda square. So therefore, if you plot as a function of delta lambda square, you get a straight line. So the circles that you see, the orange circles, are many measurements of many bits, and the statistical average over the statistical mean of these different experiments is shown as a red square with its corresponding error. So it works. So the question here I show you until now, and this is I see that I have just 3 minutes of time or something like that. So just what I told you right now is when you can measure the force totally. But in principle, many times you cannot measure, or in most experimental situations, you cannot measure the force acting on your probe. Why? Because your probe, as I'm showing here in the illustration, is interacting with a system. If it interacts with a system, then my net force is a sum of the force that I measure in my optical trap or my AFM or my magnetic tweezer or whatever your physical probe you are using, and the force exerted by the system to which your probe is in contact. And that second force you cannot measure. So what can you do in this case? What you can do then is that you must assume, and this is connect with the slide I showed you before, with an equation of a state for your system. You need some additional information of your system to apply your constraint, your law of conservation for the variances. And therefore, you need to model your non-equilibrium steady state. And this is something that you cannot avoid. Here we did for the stochastic switching trap that we mapped actually in a problem in which this force that we cannot measure is an acting force that comes from a non-equilibrium system. So what we introduce is a model, the same model as you saw before, but instead of stochastically switching the trap, an active system in contact. And you can prove that there is a reduced form of the variance sum rule that now only depends on the variances of the displacement. So the force that I cannot measure, it's not there anymore. So the idea is that if you cannot measure the force, that's not a problem. You still can apply the variance sum rule if you have a model for your non-equilibrium system, which would be an equation of a state for my gas in the language of the irreversible expansion that I was telling you before, plus the equation of the model plus just the experimental measurement of your signal, the variance of your displacement. Okay, so this reduced variance sum rule, it was applied for the stochastic switching trap, but we applied also for red blood cells.
1 minute, Felix, if you could start to finish up.
Yeah, so just to make the story short, we apply this to red blood cells. In red blood cells, you have a signal like you see here, and then what we were able to do was to introduce a simple model that describes, and this would be my equation of the state for my system, that describes the power spectrum, okay, of my system. Together with the variance sum rule, we fit everything. And what we were able to do was to measure the entropy production rate of the red blood cell across different conditions of the experimental system. And in particular, what we did was not only to measure this with optical traps, we did that also with ultra-fast microscopy. There is no optical trap. We just measure the displacement of the flickering of the membrane. And what we get is a full heat map of the red blood cell. You can find more details of this work in our paper. The result of the entropy production rate agrees with the experimental results report in bulk calorimetry. And just to end up, I want to tell you that this measurement of the red blood cell, which is about 10 to the 19 kBt per kilos per second, it coincides interestingly with what you measure in the human body. In the human body, it's known that we dissipate 1 watt per kilo, which is about 10 to the 20 kBts per kilos and second. But if you go to a ribosome, you get the same number. So ribosome dissipates about 1 kBt per second, and this has been measured in single molecule assays. So we are on the same range of 10 to the 20 kBts per second. So this calls for an homeostasis of entropy production in the non-equilibrium, in the most interesting non-equilibrium steady state, which is probably ourselves, our physical bodies. living systems. So I will stop here, thanking my collaborators. And this was mostly done by Ivan Dieterlitzky, who is now in Dresden, in collaboration with Marco Vallesi and also Marta Gironella from Barcelona. Other collaborators were Timo Betts from Grottingen, Diego Herrera from Madrid, and Paco Monroy from Madrid. I leave you here with the discussion, and sorry if I was a bit late. I hope we have some time for discussion. I can show some additional results. Thank you.